thank Jeff and Tasha and the staff of the Children's Cancer Foundation for this opportunity. I'll give an outlook of the work that we do at Children's National with specific emphasis on pediatric brainstem tumors. Okay. Some of the active research areas at the Children's National include medulloblastoma, which our colleague Dr. Brian Rood and Yanxin Chen followed, DIPG, diffuse intrinsic pontine glioma, the brainstem tumor, immunology, immunotherapy, clinical trials led by our colleagues Jin Huang, Lindsay Kilburn, and Russell Cruz, and low-grade glioma and astrocytoma by our neuropathologist, Dr. Chen. Dr. Ho. A quick background here about the DIPG. It counts for about 20% of all pediatric CNS tumors. There are between 300 to 400 cases per year in the US. The peak age of onset is between the ages of 5 to 10 years. And there is more than 95% rate of mortality. Most of children diagnosed with DIPG die within 1 year of diagnosis. So you've heard one of the story of Jake today. One of the major problems for us to do research is to have access to tumor tissue. The status for pediatric, for children diagnosed with DIPG has not changed for the past 45 years. And one of the major reasons for this not to change has been lack of specimens for us to study, lack of biological studies and lack of good preclinical models that we can test. So we recognize this at Children's National, doing DIPG research, medulloblastoma research, among other pediatric childhood, was that what are the challenges that we face at Children's? In order to understand disease biology, we need to have biological specimens. The tissue type is really important. Is it frozen? Is it formalin fixed? Is it matched? Do we have matched blood or CSF or other tissue that we can use as control? Clinical annotation of specimens is highly important also to understand pediatric diseases. Molecular annotation of the samples is also important. In order to, once we understand the biology of the tumor, we need to test hypotheses. So you've heard some of the talks today that they detect mutations or gene fusions that could or could not be leading to the cancer. We need to test those. In order to test them, we need to have preclinical models, which have been kind of mostly lacking for pediatric diseases, including DIPG. And for diseases that we do have clinical specimens, these are cells or mouse models, they're not perfectly annotated. It's been quite a while that we think of pediatric diseases as adults, specifically with cancers. And in the past few years, 5, 6 years, has been that we have a better understanding of pediatric cancers. And you hear this more often, that children are not just small adults. Their diseases are also different. We need to study them by themselves. So at Children's National, what we proposed 5 years ago was that to have a team science. And the team science includes having neurosurgery, neurology, neuro-oncology to work together with research. And the main focus was that to gather specimens. You have some of the colleagues here, Dr. Dome and Dr. Rood from Children's National, who actually helped us to make this happen. The idea was that they would work together, the neurology, neuro-oncology, surgery, and to give specimens to research. This would include tumor, non-tumor, CSF, blood, urine, any specimen from a child that had cancer. We would do the molecular testing of this. This is anything available. You heard today from Dr. Javed, who described some of the genomic, cutting-edge genomic technologies for doing this, including proteomics. Then we would take some of these samples, develop in vivo, in vitro models for our preclinical testing. This would translate into rapid application of targeting these molecules that we identify, with the aim of this feeding into our colleagues in oncology and neuro-oncology and surgery to be able to treat the children better. And I'll give you an example of what we have actually accomplished at Children's National with our collaborators. So next, besides doing collaboration and translation research at Children's, we've had also been active in collecting postmortem specimens, which for lethal pediatric tumors, including DIPGs, is highly important. Our center is one of the most active centers in collecting postmortem samples. We also do clinical studies and collaborate with other centers, including Johns Hopkins, which you've heard today from our colleagues, and the National Cancer Institute, Kathy Warren. We collected samples from all over the US and Canada to develop a biobank. This has resulted in a biobank that when I started in 2010, we had 12 samples. Now we have 868. These are patient specimens, which represent over 2,000 samples that we have. The type of samples that we have collected, the majority of them are between the ages of 0 to 5 years old, which is the majority of the cancers that we study. And they fall almost equal between the ratio of the male versus female. The type of tumors that we represent in our bank are medulloblastomas that are going into gliomas. And if you look at the gliomas that we have, the majority of them are brainstem gliomas. That again, we have either collected from biopsies or during the surgery or at autopsy. And optic pathway gliomas also are another subset of the samples that we have collected. What is good about the biobank that we have established is that we have matched samples. These include FFP. These are fixed samples that we can study. These are perfect samples for our colleagues at pathology to study. We also have CSF, blood, and urine, and you see that there is a fairly good correlation, overlap between the type of the samples that we have. Because often when we study the genetic of the disease, we need to know if this is a mutation that does exist in the normal sample as well or not. Some of this will allow us to study that. Some of the samples that we have will also allow us to study the molecules that have been shed and released by the tumor. This is called liquid biopsy. That will be explaining some of that, showing some of that data today. The biobank that we have developed has resulted in some of the publication that I will be showing here. We have been able to show that there is a protein called NG2 that seems to be driving the tumor. It keeps the DIPGs at least in the form of pluripotency. This is when the cells can divide into, these are undifferentiated cells that can divide and become into any form of cell. We've also done comprehensive multidimensional analysis with heavy emphasis on proteomics and protein profiling. Again, with the medulloblastomas and DIPGs, our center is one of the strongest places doing medulloblastoma and DIPG proteome research with our colleague, Dr. Brian Root. We're trying to, in some of the publications, we've been able to take CSF and urine from the patients with DIPG and show that they actually have, we can actually detect proteins that have been secreted from the tumor. We have also developed standardized autopsy procedures for collecting the sample. This is very important for deadly childhood cancers that we don't know much about. And we have recently looked at the spatial and temporal homogeneity and heterogeneity of the DIPG that I explained. So the question has been that if you have DIPG that grows in the brainstem and metastasizes throughout the brain, what is the nature of this tumor? You've heard today from Dr. Eberhardt about the heterogeneity of medulloblastomas, meaning that if you look at the tumor cell, if you look at the tumor mass, you look from the east to west, north to south, how does it differ? And that's very important for our colleagues to treat the patient and to treat the tumor if you want to, to understand this heterogeneity if you want to treat the tumor. So we wanted to do this for DIPGs. And because we have developed a good biobank, we took the samples that we had, again, whole brain that we collect at autopsy. And we collected over 280 samples from 8 whole brains. And we did comprehensive analysis. This is all of the omics that you've heard about today, the genomics, the proteomics, the metabolomics, and did a comprehensive comparative analysis of 380 samples throughout 9 whole brains of patients with DIPG. And some of the studies, some of the results that we have found is that while you have 100% of the tumors detected in the brainstem, you also get 25% of in the 25% of the cases you do find, you can also detect tumor in the cortex. In 29% of the cases, you can actually detect tumor cells in the frontal lobe. We've also found that if you take a histologically normal section of the brain and show to a pathologist and say, is this normal? They would say, yes, normal. If you do deep sequencing, it's normal. But if you do staining for the mutant gene, you can find about 4% of those cells are mutations. Within a perfectly normal part of the brain, you can find tumor cell migration and infiltration. However, these tumors seem to be fairly homogeneous, which is good news for treatment of the IPG. What we have identified here is that the tumor subtypes fall into two main groups: cell type regulation or growth factor signaling. We've divided this into main driver mutations and obligate partner mutations. The next slide, please. So main driver mutations and obligate partner mutation. And based on this, you can classify the IPGs into molecular subtypes that then we can assess better treatments for them. Next slide, please. So I just need the battery for the pointer. Next slide, please. And next. So we recognized two challenges, again, just to refresh. One was to study the disease. We needed biological information, which we have acquired. Then to do hypothesis testing and translate, which was our next challenge. Next slide, please. We've taken from the biopsies and autopsy samples that we've been able to develop cell lines. On the top, you see neurospheres. These are uncommitted cells that are perfect for testing preclinically in the laboratory setting. These cells do develop histone mutation, do express histone mutation, which is the hallmark of the IPG. We can also make these cells to become adherent to express GFAP and SOX2 and other stem cell markers that are expressed in gliomas. Next slide. And if you take these cells and inject them into mouse brain orthotopically, either into the brain stem, which you see in the first slide, on the left, this is the brain stem. The letter P indicates the pons. They do grow into the highly infiltrating tumor cells. Again, these are the cells that were taken post-mortem from patients, and they do express human mitochondrial antibody and also do express histone mutation. If you inject this again into the cortex of the mouse, they do also grow into the tumors. One of the current studies that we have here that I'm not showing is that we're taking tumors from the brain stem of the IPG patients and cortex and inject them vice versa into the mouse brain to see what happens. In some of the cases, what we've seen is that if you take a brain stem tumor and inject it into the cortex, the mouse dies from brain tumor, and the tumor is mainly detected in the brain stem. That says something specific about the microenvironment of the brain stem that makes it more favorable for the histone 3 mutation specifically. But we have to translate the finding in the laboratory to the clinic. And to do that, we have teamed up with a group of Pacific Pediatric Neuro-Oncology Consortium, PNOC. I know we are on the East Coast, but we are a part of the West Coast Pacific Pediatric Consortium, which is mainly run by our colleague Sabine Mueller at UCSF. This is the general scheme of the study that we have. The patients who are diagnosed with DIPG, actually this study was recently funded to include the low-grade glioma as well. They are enrolled into the study. Upfront biopsies are collected. We do genomic analysis of the biopsy within 2 weeks. The genomic analysis, this is the exome sequencing and also RNA sequencing, is then reviewed. We also take a section of the core biopsy that we take to develop xenograft and cell culture that we can study. Then based on genomic pattern of that patient, the patient is put on specific treatment, specific drugs that will target the mutation. This is the general scheme of what we do. The biopsy, we make in vitro and in vivo models, and a second biopsy is also taken at recurrence when the tumor grows if it does not respond to treatment. We also study the biopsy in vivo and in vitro to see what are the mechanisms of resistance. The idea is that we would compare, this is called trans-species comparison. We have the genomic data from the patient, we have the genomic data from the mouse. We do put the mouse under the same treatment that the human has gone through, the patient has gone through, and we do compare to look at resistance to the tumor. So if you look at the initial data that we have collected through the PNOC study, we show that the PNOC patient, the upfront biopsies that we've collected, the genomic aberration of those that overlap with the post-mortem sample that we had analyzed. The study that I showed you before with the driver mutation and their obligate partner mutations is exactly replicated at upfront. This showed that the mutations that we see at post-mortem, they existed when the tumor was originally detected, before any type of treatment. If we want to treat the tumor, we have to go by the mutations that we detect at post-mortem. So an example of what we have done with mouse and primary cell is that, next, for one of the PNOC patients, for example, we injected the biopsy, one of the core biopsies into the mouse, and then we cultured those. Within 8 months, the mouse died. The mouse developed the same tumor that we have taken subsequently and have done genomic analysis and injected into another mouse. Another example is that a patient that we took biopsy on March of last year, the tumor was developed within 5 months, and we injected into the mouse, and 4 mice have been subsequently re-injected with the biopsy that we took from the patient. One of the complications that we have in patients is that if you have 2 different mutations, and for the 2 different mutations there are 4 different drugs, how would you go about which drug would you select to target to treat a patient? In the mouse models that we have, they carry exact same mutations. We could do that with mouse because the mouse is carrying the same genetic alterations. We can have different mouse cohorts and put them on different combination of mutation and assess response to treatment. Which is another example. This is a simplified example that you're looking at 2 different mutations, p53 having 3 different options of treatment. So we match and mix different options and look at the outcome of the therapies. This is an example of how complex it would be if you want to target all different mutations and what would be the potentially different drugs that you could pick. Or in mouse we could do it, but in human we cannot. So one of the complications that we have in the clinic is that the drug delivery. Some of the drugs that show response in the cell, they would never make it. They may even look good in mouse because we're doing orthotopic injection. We're actually penetrating the blood-brain barrier. The drug may be getting into the tumor because we have already altered the blood-brain barrier. In patients, they may not be able to get into the system. One of the approaches has been to do convection-enhanced delivery, is where the surgeon will put the catheter into the tumor directly and directly infuse the drug. We want to do this in the laboratory. But some of the question is that what is the injection rate, for example? Would you be able, how much of this can you get into the tumor if you have a slower rate of the same amount versus a higher rate? This is an example of what we have done with just injecting dye at 2 different rates, the same amount of dye at 2 different rates. The optimal rate that we have found is to be 0.3 microliter for our models that had a more focal delivery of the drugs into a smaller area of the brain, which is the tumor region. We can also see this in live animal imaging. If we inject them with the dye that we can detect in the live animal imaging, that a smaller rate of the same volume of the drug can actually be more focally delivered to the brain. And this is a quantification of different rates that I just mentioned. But also the idea is that if you have the tumor that is growing in the mouse, you want to go back to the same tumor, deliver this to the exact same spot. We're using the guide screws, which allow us this screw allows us to inject the tumor through the hole that is in the screw. And then once the tumor forms, we go back to the same spot, we re-inject it within the same tumor to make sure that we're delivering to the site of the tumor. With respect to plasma and CSF, one of the problems that is in the clinic is that is the patient responding to the treatment or not? An assessment of the response is typically done radiographically, and radiographic assessment is not always specific. We've tried to develop a liquid biopsy platform where we take plasma and serum from patients and we try to see if we can detect tumor-specific mutations. In this case, we've taken CSF and serum and plasma from patients with DIPG, and we look to see if we can find histone mutation. The idea is that the tumor cells that are growing in the brain stem, they will shed the histone mutant genes, and we can detect them in the serum and plasma. In this example, in the bottom panel is a control, healthy control, and the top panel you have the serum of the patient with DIPG. WT is the wild-type histone. The mutant is the mutant histone that was detected in the serum of the patient. Again, this is an archival sample that we've had stored for 5 years, and we tested to see if the platform works. This is a digital droplet PCR that is very, very sensitive for detecting this mutation. In this case, we can also detect this in the CSF. This is a very good method of detecting. We've actually got this very sensitive down to detecting 10 copies of the mutant gene in the serum of patients with DIPG. The idea is that what I presented to you early on is that to have the neurology, neurosurgery, neuro-oncology to work with the research, with the idea of having a feedback to them such that they can treat our children better. The progress that we've had over past 5 years to go from the laboratory to the clinic to provide more information to our colleagues in the neurosurgery and neuro-oncology to treat children, and our colleagues at PNOC, this has been a great example of it. They have more information to treat their children. And hopefully with the PNOC trials that we have and the molecular studies that we have, we can have a more informed way of treating children with DIPG. With that, I would like to thank our patients and their families for making everything that we have done possible, and also funding foundations. So we are 100% funded by foundations, as you can see, which is the case with most of childhood cancer researchers. And thank you.
We have time for a couple questions. Okay, everyone's hungry.
Yes.
Thanks, Javed. Very nice overview. Can you tell me more about the liquid biopsies that you presented at the end of the talk? Do you have to sequence the tumor first and then establish that assay based on the mutations that are found in the tumor? And what's the turnaround time on that?
No, so if you know what are some of the mutations, if you know the mutation of the tumor, you can take serum and see if you can detect it in the serum or the CSF. The most difficult part of this is to develop a specific probe that can detect that particular single nucleotide change, because the probe has to work for both mutant and also the wild type sequence. And it will take, for us to detect histone mutation, it took about 2 months to perfect the methodology. The temperature, the right probe. We went through with 3 different probes to be able to detect histone. But we have a platform now. We have a panel that we can detect 2 different histones, histone 3.3 and histone 3.1, with the idea of developing it into with the exception to the P53, which has multiple mutations, to all of mutations that are detected in the IPGs. Yes.
So, very nice work and presentation, and you've done an amazing job, you and your team, of picking one of the huge challenges in pediatric oncology and coming up with systemic and ways of you have to not only find creative ways to get the tissue, which you've done, but now you're going to have to analyze it. And so the next challenge, of course, is going to be drugs. How do you make drugs for these? And based on these mutations, I know that that's not going to be so easy.
Right.
So it's the next challenge. But I wanted to ask you about what you have done so far. It looked like in the 9 DIPGs that you did sequencing on, that you showed, that you did multiple samples from each of those, and that there was relative homogeneity. And there was a few exceptions, but to my eye, as you put the slide up, it looked like there was a tremendous amount of homogeneity, which is actually very interesting because some people would argue that cancer is a problem of heterogeneity. But in your case, homogeneity. Can you comment on that?
Yes, well, thank you for a comment. So I think when we talk about cancer homogeneity versus heterogeneity, there are two different views of this. There's some work coming out of Chris Jones' lab showing that the IPGs are actually heterogeneous. If you take any tumor sample and you take 2 different cells and do deep sequencing, they're definitely heterogeneous. Even any normal sample, if you take 2 genomes, the expression level of the RNA will be heterogeneous. For DIPGs, what we have proposed is that the tumor picks up one mutation first, and that's a driver mutation and an obligate partner mutation, and stays with these 2 obligate partner mutations throughout the tumor. Anywhere in the brain that you detect this, it has both of this obligate partner mutations present in them. It never loses one or another. The only time that we have seen a tumor that picked histone mutation and p53 and lost p53 is when it lost p53 and picked PPM1D, which run the same pathway. The pathway that drive tumorigenicity always stay the same. The obligate partner mutations, they always stay the same. However, tumors are heterogeneous if you look at the different mutations across the sample. If you're thinking about targeting a tumor, the IPGs are homogeneous with regards to their obligate partner mutations.
Any more questions?